We will continue the biochemistry. Biochemistry I'll teach slowly, okay. One second. Okay, so as you all know, in the entire syllabus, biochemistry is one of the toughest unit, and we get the questions also very tough. Okay, but remember, for the exam point of view, in order to score a good number of questions in the exam, I want every one of you to become expert in few topics, whatever I am writing here. In that, number one is the complete carbohydrate metabolism. A to Z we have to read in the carbohydrate metabolism, A to Z, okay. Starting from glycolysis, glycogenesis, genolysis, gluconeogenesis, we'll see that. Then after the carbohydrate metabolism, the must read topic is amino acids. I don't know why your voice is low. Okay, the next topic is amino acids. Okay, then the topic where we get the questions more oftenly, that is proteins. Whenever we are reading the proteins, we have to read every aspect. So such as we have to read protein structure, we have to read protein folding, we have to read Ramachandran plot, we have to read protein denaturation, we have to read protein kinetics. In simple words, we have to read every aspect of the protein. Then apart from the proteins, see, these three are compulsory. In every single question paper we get the questions from there. Then the fourth one, we have to focus on enzymes and its kinetics. Whomever trying to crack at this December exam, anyway I will teach the whole concept, but remember, you give highest importance to these four subtopics, okay. But if you give importance to only these four subtopics, also difficult. If a very difficult question appear from here, and if other easy questions coming from other units, so you may lose the marks. That's why read the whole lesson, but give the highest importance to these four subtopics. Definitely you get 4 to 6 questions only from these four topics, definitely, okay. Now again, I'm just telling you one word. Biochemistry is all about intelligence rather than reading, rather than remembering, okay. Definitely we have to remember a lot of information, but before remembering the information, we have to understand. With a thorough understanding, without reading only, we can give the answers to the questions, okay. Hence focus on concept majorly, okay. Now so first we will be starting about carbohydrate metabolism. In the carbohydrate metabolism, we have to read various steps. We have to focus on glycolysis, we have to focus on Krebs cycle, we have to focus on electron transport chain, we have to focus on oxidative phosphorylation, right, and we have to focus on PPP pathway, pentose phosphate pathway. Then we have to focus on glycogenesis, that is synthesis of glycogen. Then glycogenolysis, breakdown of glycogen. Then gluconeogenesis, okay. And another important one, shuttle systems, okay. So all these 9 subtopics we have to read very briefly, and trust me, all these 9 subtopics, 90% you have to understand, 10% you have to remember the information. That's all, nothing else to do it, right, okay. So now the most important thing, how the questions can be framed. 90% I can say that the question never come directly, question always come indirectly, and that too in the form of a application based only, okay. So only focus on the concept, don't focus on remembering, okay. Now metabolism, we have to read carbohydrate metabolism, protein metabolism, everything, okay. So simply understand what is called metabolism. Metabolism is a series of chemical reaction which includes catabolism and anabolism. Suppose example, when we take a food, consider just now I had a food, the food contains carbohydrates, proteins and lipids. Ultimately, why we take the food? In order to produce energy. How can we produce the energy? By breaking the food, correct na? So what is catabolism? It is a oxidation or breaking of biomolecules. What are biomolecules? Carbohydrates, proteins and lipids. So catabolism is a process of breaking down or oxidation of biomolecule. Why we are breaking down? To produce energy. What is universal energy currency? ATP. So we all know very well, in a typical human body we have 37.2 trillion cells, not billion, trillion cells. Every single cell exclusively depends on the ATP in order to perform all the function. Consider it is a muscle movement or a hormone secretion or an enzymatic activity, everything we require ATP. So ATP will be used in what purpose? Anabolic function. Anabolism is also called building up reactions. Means the reaction where there is a requirement of ATP. Correct na? So I am using the ATP. So can I say that in the anabolism ATP hydrolysis occur? Correct or not? I am using the ATP in the, see, suppose I want to move a muscle. Here what I am using? I am using energy. Then definitely ATP break down into ADP plus PI. Correct na? So most of the students get confused that catabolism means what? Breaking down. So everyone think that breaking down means we are breaking the ATP. We never break. Catabolism, we produce ATP. By breaking what? Biomolecules. By breaking carbohydrates, protein, lipids, we produce ATP. This ATP can be hydrolyzed or break down while performing any function. So a series of catabolism and anabolism together called as metabolism. Correct na? Don't confuse. Now, so this is the most important slide everyone understand. So just now in the plant physiology we read something called as a production of the glucose by photosynthesis. However, plants are producing the glucose. We, carnivores, we will eat plants and we will also get the glucose. Now what happens to that particular glucose? What happens to the proteins that we take? What happens to the lipids that we take? So generally lipid has to undergo catabolism or breakdown. Polysaccharide has to undergo the catabolism. Proteins also has to undergo the catabolism. What is ultimate goal? Why we are breaking all this? We are breaking all these ultimately to get energy. Correct? Now that's the reason we eat the food, to get the energy to perform all the body function. So first let's understand the breakdown. This is the slide of overview, the most important slide. Focus. If you understand this slide, most of the concept is very clear. So first, we never consume the food directly in the form of monosaccharides. Mostly we consume the food when it comes to the carbohydrates in the form of polysaccharide. Let's consider we are eating a rice. Rice is what? Generally starch. Starch is generally polysaccharide. So we take the food or the carbohydrates in the form of polysaccharide, but finally it is breaking down into monosaccharide, because if you consider the brain never take the energy from polysaccharide. Brain always utilize the energy from glucose, which is the simple sugar. So whether it is a polysaccharide or a disaccharide or oligosaccharide, finally it has to break down into what? Monosaccharide, which is glucose. Now finally we will see that this glucose undergoes what process? Glycolysis process, and giving rise to pyruvate. And what happens to this pyruvate? Now very important, pyruvate entered into the mitochondria and converted into acetyl-CoA. Acetyl-CoA enters into which cycle? Citric acid cycle, produce abundant amount of ATP. Our ultimate goal is to produce ATP. So carbohydrates such as polysaccharides finally convert into glucose. Glucose undergoes glycolysis, convert into pyruvate. Pyruvate convert into acetyl-CoA. Finally acetyl-CoA enter into the citric acid cycle and producing ATP. So what I am trying to say, this is the fate of a polysaccharide in our body. Ultimate goal is to produce energy only. Along with the polysaccharide, we definitely take the lipids. Correct na? Lipids also has to break down, right? Lipids break down into fatty acid plus glycerol. Correct na? So now I understand the point correctly. Glucose converted into pyruvate by the mechanism called as glycolysis. Similarly, fatty acids are also undergo the breakdown or catabolism. The catabolism of fatty acid is called as beta oxidation. Now, upon beta oxidation, fatty acids are also giving rise to Acetyl-CoA enter into the citric acid cycle, produce ATP. Similarly, lipids can also give rise to glycerol. Now the glycerol converted into pyruvate, pyruvate anyway giving rise to acetyl-CoA, the same cycle will be repeated. So in simple words, lipids are also ultimately giving rise to what? Acetyl-CoA, right? Enter into the citric acid cycle. Same goes with the protein. When we take a protein, protein is a very large molecule, break down into simple amino acids. Now one amino acid can be converted into another amino acid by a mechanism called as transamination. Upon transamination, amino acids are releasing pyruvate and again they enter into the citric acid cycle, produce abundant amount of the ATP. Now whenever the transamination reaction is happening, there is a release of what? Ammonia and urea, because they should not be stored, they are extremely toxic in nature, so they are definitely eliminated through the urine, right? Now what's the common statement we can understand? Whether it is a lipid, whether it is a polysaccharide, whether it is a protein, all the biomolecules are finally giving rise to acetyl-CoA that enter into the citric acid cycle and produce abundant amount of the ATP. So the common question in the exam, which of the following can act as a key intermediate in the metabolism of biomolecules? Which is acetyl-CoA. Because whether it's a protein or a lipid or a polysaccharide, if it wants to produce the energy, they have to enter into the citric acid cycle definitely. And they enter into the citric acid cycle only with the help of acetyl-CoA. That's the only reason this is said to be the most important key intermediate. Okay, now there is a very simple question. So what are the biomolecules we can see here? We can see protein, we can see polysaccharide, lipid. I am adding here something called DNA, and I am also adding RNA, and I am also adding cellulose. Okay, so 1, 2, 3, 4, 5, 6. Among these 6, which is not a polymer? This is the neat question of intermediate. Okay. Huh? Everyone online people? Which is not a polymer? First of all, what is polymer? Vagdevi, what is the answer? Who are son? I'll send all of you to 10th. Huh? This question also you can't answer. What is polymer? Very good. Polymer formed from many monomers. Simple. Protein made up of what? So definitely protein is polymer made up of monomers. Correct na? Polysaccharide. Take an example of polysaccharide. Starch. And I have given one example now. Cellular. Cellulose is generally made up of what? Glucose. Many glucoses combined together and giving rise to the cellulose. So cellulose is a polysaccharide, nothing but polymer made up of monomers, which is the glucose. DNA made up of nucleotide. So one nucleotide is one monomer. Many nucleotides giving rise to the DNA and RNA. RNA and DNA are definitely polymers. What about the lipid? Lipid is not a polymer because it is formed by the condensation of glycerol and fatty acid. Correct na? There are two different substances giving rise to the lipid. That's why it is not polymeric in nature. Okay, no one is answering. Okay, not a problem. Okay, now what exactly we have to focus now? First, we have to understand whether it is a protein, polysaccharide or lipid. Finally, they are going to give rise to the acetyl-CoA. Correct na? First, we have to understand how does this glucose giving rise to the pyruvate and how this pyruvate is giving rise to the acetyl-CoA. After that, we will understand all the concepts very clearly. Okay. One second. Okay. Now, throughout the biochemistry, we will always come across about these three terms: ATP, NADH, and FADH. Correct na? So it is important to know about them. Okay. So what is ATP? It is a universal energy currency cell. Correct na? You observe the structure of the ATP. How does it look like? It contains a sugar. What sugar it is? Ribose sugar. Correct na? And it contains one base. What base? Adenine. And it contains 3 phosphate. we called alpha phosphate, beta phosphate or gamma phosphate or mono di triphosphate. Correct na? This is the simple structure of what? ATP. How do we get ATP? Just now we discussed now by the oxidation. Correct na? By breaking down or catabolism of the biomolecules we get the ATP. Correct na? Now the most important statement to remember, see, when I break one ATP molecule, okay, slowly understand. When I break one ATP molecule, I can't break directly into AMP. First I can break down into ADP plus PI. Then can I break ADP back to AMP plus PI? Correct na? Similarly, in opposite direction, AMP combines with inorganic phosphate give rise to ADP. ADP combines with inorganic phosphate give rise to the ATP. Correct na? So, remember correctly, it requires nearly 7.3 kilocalories of energy will be released out when one ATP break down into one ADP. When ADP break down into AMP also, there is a release of 7.3 kilocalorie mole of energy. So, in very simple words, when one ATP breaks down into one AMP, we get nearly 14.6 kilocalories of energy. Okay, compulsory remember that when one ATP break down into the AMP. Similarly, if I want to make the ATP from the AMP also, I require 14.6 kilocalories. Correct na? There is no point of getting confused over there. Okay, slowly answer, don't be hurry. I'll throw all of you out if you make wrong. Okay, remember that word. Okay, simple. Everyone online? Why no one answered that simple question? Online people. You have to choose 1, 2, 3 like that. Sorry, only two options are there. 1, 2. What's the first option used in anabolic reaction? Is it ATP or ADP? AT? 1 or 2? 2, okay. Now understand correctly. How can we use ADP in the anabolic reaction? When we use ATP, that breaks down into ADP. When I use ATP, then I'll break it into simple pieces. Correct na? So we can't use ADP. ADP was a byproduct, released product. Hence it is 1. The energy storage molecule, which is also an ATP. Now slowly understand option C. Coupled with energy requiring reactions. What does that mean? Just now we discussed an anabolic reaction, the reaction which requires ATP. That is called energy requiring reaction. If I told you right, if I want to move a muscle, this is what reaction? Energy requiring reaction. So definitely what I required? ATP. Correct na? When ATP break down, we get ADP and PI. So these are said to be what products? Hydrolysis products. Correct na? So understand that correctly. Okay. So that is a simple structure of the ATP. Okay. So just give me a minute. Just a minute. Just It's an important image. Where is that image? Just a moment. 1 second, I'll just show it. Small image got missed. It's okay, no problem, I'll write with hand. It's okay, it's okay, continue. So, we understood very clearly that, first carbohydrates has to undergo the process of digestion, and then finally they are going to give rise to the glucose, correct na? So now this glucose undergoes variety of reactions and finally it is going to give rise to the ATP. So in very simple words, metabolism of carbohydrates always starts with what? Glucose, or the oxidation or breakdown of the glucose. So in the breakdown of the glucose, the first step is what? Glycolysis. Now remember the line, glycolysis is an ancient and linear metabolic pathway. What about the Krebs cycle? Krebs cycle is always circular pathway, it is completely a linear pathway. And the very important statement, glycolysis will happen in every single organism. And where is the site of the location of the glycolysis? In the cytosol of every cell in our body. So what's the ultimate goal of the glycolysis? All of you focus on the picture. We are breaking down one glucose. One glucose is how many carbon? 6 carbon. I am breaking into 2 molecules of pyruvate. Each pyruvate is how many carbon? 3 carbon. So one glucose giving rise to 2 pyruvates, 2 NADH, 2 ATP, and loss of or release of 2 water molecules. So always remember the same equation. 1 glucose, 2 pyruvate, 2 NADH, 2 ATP and 2 water molecule. Then finally this pyruvate enter into the mitochondria, convert into the acetyl CoA. Correct na? So now this glucose never converted into the pyruvate in single step. It takes multiple steps. It is a series of 10 enzymatic reaction where the glucose can be converted into what? Pyruvate molecule. Correct na? Now everyone just try to understand the concept very correctly without confusion. This is a very important step. What is the first step? You already read. Glucose is converted into glucose 6-phosphate, which you know it. Now the question is here, why in the first step glucose has to be converted into the glucose 6-phosphate? Why glucose cannot convert it into the fructose or into something else? Why only glucose has to be converted into the glucose 6-phosphate? The reason, when the glucose converted into glucose 6-phosphate, phosphate is what charged? Negatively charged. So negatively charged molecule cannot cross the plasma membrane. Hence, it will be located within the cell itself. Then in the cell, it undergoes variety of chemical reaction. Suppose example, if the glucose enter the cell, there is a possibility that again it leave the cell. So to protect the glucose within the cell, we will convert the glucose to glucose 6-phosphate. This mechanism is called as glucose activation. This is a very important line to be remembered. Okay, now focus. Now all of you answer. Now every one of you focus from this slide. Whatever we discussed is small basics. Now everyone focus from here. Now all of you please give me a simple difference between cellular respiration and common respiration. What is the difference? Respiration, exchange of gases, inhalation of oxygen and exhalation of carbon dioxide. What is called cellular respiration? Cellular respiration is a mechanism of oxidation of biomolecules to produce energy. So by using cellular respiration, we oxidize all the biomolecules and finally we produce ATP. So in the cellular respiration, there are multiple steps are involved. The number one step is what? Glycolysis. And citric acid cycle, electron transport chain, oxidative phosphorylation is the fourth step. Now why this many steps has to happen? What is our ultimate goal? See, by breaking down the polysaccharide, what we obtained? Glucose. Now what is my goal or goal of the cellular respiration? By using one glucose molecule, we have to produce 36 to 38 ATP molecule. This is our ultimate goal, because we discussed that when we break down the food, we get energy. So by breaking down of one glucose, we get this much of ATP. But you cannot produce this much of ATP in just one step of the glycolysis. So for that, there are multiple steps are involved. Through all these steps, one glucose molecule undergo the mechanism of oxidation and produce 36 to 38 molecules of ATP. Right. And it is very important for the exam, we have to remember the locations. We already discussed it just now. Glycolysis is strictly happening in the, because all the 10 enzymes are located only in the cytosol. Now glucose is going to give rise to pyruvate. Correct na? And we will discuss, but we already discussed it last time. Now pyruvate is entering where? Where? Mitochondria. Mitochondria having how many membranes? Outer membrane and inner membrane. As you all know very well, outer membrane is permeable. We read in the cell biology. Inner membrane is impermeable. Now my question is, how pyruvate can enter into the mitochondria? By using certain transporters. We call pyruvate translocase. We'll see that. Then pyruvate enter into the mitochondria and giving rise to acetyl-CoA. Now acetyl-CoA enters into the, and where the location of the citric acid cycle? Mitochondrial matrix. Why only matrix? Because all the enzymes of the citric acid cycle is located in the matrix. Now, whenever the citric acid cycle over, what do we get? We get ATP, we also get NADH, we also get FADH. What is the difference between these three? This is the direct energy, you can see it. In this also there is abundant amount of energy which we can't see. Correct na? So, and morning class we read multiple times, NADH to also act like electron carrier. So whatever the NADH and FADH produced in the Krebs cycle, they carry the electrons to where? Electron transport chain, which is located in the inner membrane of mitochondria. There NADH and FADH to finally going to give rise to the ATP by a process called as chemiosmosis. So if one glucose molecule wants to give rise to 36 to 38 molecules of ATP, definitely what do we require? We require 4 steps. In one step we can't produce, right? So all the 4 steps together called as cellular respiration, means oxidation of glucose to give rise to bulk amount of ATP through these 4 steps. Okay, the first and most simple step is the glycolysis. You can see, right, in the glycolysis what is happening? One 6-carbon containing compound is going to give rise to 2 pyruvate. Each one is how many carbon? 3 carbon. It is a so simple step. Finally pyruvate is going to convert it into the acetyl-CoA. Now let's understand how does it happen. Okay, before that I want to highlight one very important slide. Focus. Understand, more than 6-7 questions were appeared in the same way. Okay, so first glucose undergoes what pathway? Glycolysis, and finally giving rise to pyruvate. Correct na? We will discuss that. This pyruvate, how many carbon? 3 carbon. If there is a presence of oxygen, what? Presence of oxygen, then only friends pyruvate can enter into the mitochondria. Very good. And converted into acetyl-CoA. And you know very well acetyl-CoA continue the Krebs cycle and produce bulk amount of ATP. Imagine there is no oxygen. Then pyruvate is how many carbon? 3 carbon. We will see that later. Now in the absence of oxygen, pyruvate converted into ethanol or ethyl alcohol. Where, in which organism? Yeast. Correct? Now only in the absence of oxygen. Same pyruvate in the absence of oxygen in our muscles, it is converted into lactate or lactic acid. Where it is happening, we'll see that this is also do not require oxygen. So this is the simple fate of glucose. What's happening here? Glucose first enter into the glycolysis in the cytosol, converted into the pyruvate. If oxygen is present, pyruvate converted into acetyl-CoA, we get abundant amount of energy. If it is absent, especially in our muscles, pyruvate converted into lactate. Correct na? This is the simple fate of a glucose in a unidirectional way. Correct na? Now imagine we just had a biryani. Consider, then we get abundant amount of the glucose. Correct na? But cells do not require cells do not require abundant amount of glucose. How much ever is required, it will use only that much. What about the remaining glucose? See, how much ever glucose is required, that glucose converted into pyruvate. Remaining glucose has to be converted. Correct na? And it is stored where? Stored in the form of glycogen. Where the glycogen can be stored? In the liver, muscle, and very little quantity in the brain. Mostly it's stored in the brain. Now don't confuse, I am converting only excess glucose into glycogen. Correct na? So what I am synthesizing? Glycogen. Genesis, called as synthesis. What I am synthesizing? Glycogen. That's why we call glycogenesis. Synthesis of glycogen. Correct na? When I will synthesize the glycogen? When I am having only excess glucose. Because today I had lot of food, I have abundant amount of glucose, that's why I converted into the glycogen. So tomorrow I am doing fasting, I don't have any energy in my body, even though I don't eat food from morning to evening, I can survive very easily. What's happening there? Whatever the stored glycogen in the liver converted back to glucose. So what I am doing, I am breaking of glycogen, lysis breakdown, and glycogen. So breakdown of the glycogen is called as glycogenolysis. When it will happen? Only whenever there is a low glucose level in the body. Correct na? Or fasting condition. So tomorrow I am doing fasting, right? And imagine day after tomorrow also I am continuing the fasting. Then that time, see, whenever we are doing the fasting up to 18 hours, we generally use the word fasting. If we continue it beyond 18 hours, we use the word harvesting. Okay, starvation is up to 18 hours. Okay, if it is prolonged, we call harvesting condition. Now, you imagine in the first day what I used? Glycogen. But in the second day also I am continuing the fasting, but still I can survive. Correct na? Why? Because now glycogen, whatever I stored, that is also over. Correct na? Now the most important statement here, whenever the glucose is excess, we are converting into what? Where it is stored? Liver. Entire glucose cannot be converted into the glycogen. Until the glycogen storages are filled, glucose can be converted into the glycogen. Then still glucose is left over. What do we do that? We convert the glucose into lipids. Correct, or fatty acid. We call it as lipogenesis or lipogenesis. Correct na? So first excess glucose we convert into glycogen. If the stores are completely filled, excess glucose we convert into the lipids. So whenever we are doing the fasting, whatever the glycogen that got stored, I converted into the glucose by the mechanism of glycogenolysis. Now next day also I am continuing the fasting, and whatever the glycogen I stored, that is also over. Correct na? But still I can survive. Why? I can get the glucose from the lipid. Lipid is a carbohydrate? No. Correct na? So this process we call it as gluconeogenesis. What is called genesis? Synthesis or production of glucose other than carbohydrate, which is called new. New represents other than the carbohydrate. Example, lipids, lactic acid, all these, amino acids, proteins. So production of the glucose other than the carbohydrates is called as gluconeogenesis. Clear? Now the most important question here. Now I am having abundant glucose in my body, the excess glucose is converted into glycogen. Correct na? So who will convert this excess glucose to glycogen? Who will convert which hormone? Insulin. Correct na? Insulin will be released in the body once the glucose levels are high. Then immediately it converts excess glucose to glycogen. By that it controls the level of glucose. So insulin only is the one which converts the glucose to glycogen. Insulin is the only one which converts the glucose to lipids also. Insulin is always involved in the genesis function, synthesis. So tomorrow I am doing fasting, I don't have any glucose in my body, then what I have to do it? I have to break down the glycogen into glucose. This can be done by glucagon. That's why insulin and glucagon are antagonistic to each other. Both are performing opposite function. Correct na? So don't confuse. Glucagon is releasing when the glucose level is lower. Correct na? So then it increases the glucose levels. Correct na? What about the insulin? When the glucose level is excessive, it is going to control the glucose level by converting into a stored form. Right? So this is important slide we have to remember. Okay. Now let's focus on our first step. After that we will discuss all these steps, that is conversion of glucose to the pyruvate. Okay, so as we discussed, conversion of the glucose to the pyruvate happens in total 10 steps. Don't confuse. First 1 to 5 steps are said to be the preparatory phase. 6 to 10 steps are said to be the payoff phase. Sometimes instead of using the word preparatory phase, we may use the word investment phase. Suppose I want to do a business. First what I require? Capital. So first what I will do? I will invest money. Correct na? Now once business is growing well, then I will get my investment back along with profit. Correct na? Now what's the ultimate goal of the glycolysis? What we have to produce? ATP. Correct na? So in order to produce ATP, I have to spend ATP. You're getting the point. That's why we called as preparatory phase. We'll spend ATP and we'll get that ATP along with the profit. We'll see that. Now, is it necessary to remember the structure? It is mandatory, okay, to remember the structures of glycolysis, structures of Krebs cycle, structures of amino acid. If you miss this, you will lose the question definitely, okay, and that is not difficult also, remember. So the first 5 steps, energy investing phase. In the first step, glucose is converted into what? Glucose 6-phosphate. Glucose is how many carbon? And glucose 6-phosphate is also 6 carbon. There is no change in the carbon number. What happened? The phosphate group is added at the 6th carbon. Correct na? So if phosphate group is added, definitely someone has to donate that phosphate group. Who is going to donate? ATP. So our ultimate goal is to produce ATP only, but we are investing ATP. Correct na? That's why it is called glucose is converted into glucose 6-phosphate by spending how many ATP? 1. Only 1. Now glucose 6-phosphate converted into fructose 6-phosphate. How many carbon it is? 6 carbon. There is no change in the carbon number. It is just converted from one isomer to the another isomer. Then fructose 6-phosphate is how many carbon?
6
Converted into fructose 1,6-bisphosphate. This is how many carbon? Still it is also 6 carbon. But what happened? In the fructose 6-phosphate, only the 6th carbon added by the phosphate. But in the fructose 1,6-bisphosphate, along with the 6th carbon, even 1st carbon also added by the phosphate. Since we added one phosphate, someone has to donate that phosphate. Again, the donor is ATP. Now the most important, this fructose 1,6-bisphosphate, which is a highly unstable compound, breaking down into 2 individual compounds. This is how many carbon? 6. Definitely it has to break down into 3 carbon. One is called G3P. Where we read G3P? Morning we read now in the C3 cycle, the stable product, G3P. Glyceraldehyde 3-phosphate. Another one is called as DHAP, dihydroxyacetone phosphate. They are said to be trioses, which are 3 carbon containing compound. So 6 carbon, how many phosphates are there? 2 phosphate. Now it is breaking down into 3 carbon with 1 phosphate and 3 carbon with 1 phosphate. Correct na? Now it is super important to remember the structures. Okay. Now this is the structure of glucose. What is this ring called as? Remember correctly? See, ribose we read, right? DNA contain what ring? Ribose sugar and deoxyribose sugar. Ribose is how many carbon? 5 carbon. Correct na? Ribose is how many carbon? 5. 1, 2, 3, 4. Remember always, 5th carbon is here in the arm position. What is that? Arm. So 1, 2, 3, 4 carbons in the ring with 1 oxygen. 4 carbons with 1 oxygen. We call it as what? Furan. What is that? Furan. Since it's a sugar, we call furanose. So ribose is what sugar? Furanose. Now look into the glucose. How many carbons are there in the glucose? 6 carbon. 1, 2, 3, 4. This is 5th. And where is the 6th one? Here. Outside the ring. It is in the arm position. So it cannot be considered within the ring. So in the ring, how many carbons are there? 5. With 1 oxygen. 5 carbons with 1 oxygen is said to be pyron. Since it is a sugar, we called ose. Pyranose. Okay. Then the glucose is what sugar? Pyranose sugar. Don't confuse. Now what's happening here now? Glucose is converted into glucose 6-phosphate. Correct na? Now don't confuse here. We are adding the phosphate group exactly at which carbon? 6th carbon. We already read multiple times. Kinase is the enzyme which add phosphate group. And what's the function of phosphatase? Which remove the phosphate group. Now what we are doing here, since we are adding the phosphate, we require an enzyme called as kinase. Glucose is how many carbon? 6 carbon. 6 carbon containing sugar called as hexose. That's why name of the enzyme is hexokinase. So glucose converted into the glucose 6-phosphate. Now observe the point. Glucose 6-phosphate converted into what? Fructose 6-phosphate. Correct na? This is the glucose 6-phosphate converted into the fructose 6-phosphate. Just there is an addition of CH2OH group. Both glucose and fructose are isomeric in nature. Now remember, we know, right, one isomer we are converting into the another isomer. Definitely the enzyme we use is isomerase enzyme. Simple. Now fructose 6-phosphate having how many carbon? 6 carbon with 1 phosphate. Phosphate is at which carbon? 6th carbon. Now fructose 6-phosphate converted into fructose 1,6-bisphosphate. So where we added the phosphate now? To the 1st carbon. Now the most important principal enzyme, this is the enzyme which controls the entire glycolysis, phosphofructokinase 1. Correct? Huh? Phosphofructokinase 1. Since it is a kinase. Correct? Na? Adding the phosphate to where? Fructose. And what group it is adding? Phosphate group. That's why we call phosphofructokinase. Right? So which add, what it is adding? Phosphate group. By that fructose 6-phosphate converted into fructose 1,6-bisphosphate. Perfect. Now fructose 1,6-bisphosphate is how many carbon? 6 carbon with 2 phosphate groups. Okay. Now the fructose 6-phosphate, which is completely unstable compound, which is a 6 carbon with the 2 phosphates, is breaking down. Remember again, whenever you heard the name of breaking is happening, always remember the enzyme involved is aldolase. What we are breaking? Fructose 1,6-bisphosphate. So the enzyme is fructose 1,6-bisphosphate aldolase, which is breaking into 2,3 carbons. Correct na? Into 2,3 carbons. So 3 carbons with 1 phosphate and 3 carbons with another phosphate. Now you only tell me, all of you, what is the difference between the glyceraldehyde 3-phosphate and DHAP? Very, very good. One is aldehyde group and one is ketone group. It is a ketose sugar, it is a aldose sugar. Now finally we completed 5 steps. Now the most interesting point here, these two can act like isomeric, but they are interconvertible. What is the meaning of interconvertible? In very simple words, whatever the function performed by one molecule, same function automatically performed by the other as well. Suppose example, glyceraldehyde 3-phosphate lost 1 carbon. Automatically it also lose 1 carbon. Suppose glyceraldehyde 3-phosphate gain 1 ATP. It also automatically gains 1 ATP. So while writing, we will write only for one molecule. But while calculating, we will calculate for both molecules because both are performing the same function. So we completed first 5 steps, which is called investment phase. How much we invested? On the first step we invested 1 ATP. On the third step also we invested 1 ATP. So in the step 1 and step 3 together we invested 2 ATP. Right. Now the 6th step. What we discussed in the 6th step onwards, we will consider only for one, but we will calculate for both. Now glyceraldehyde 3-phosphate, how many carbon it is? 3 carbon, very good. It is converted into 1,3-bisphosphoglycerate. What is that? 1,3-bisphosphoglycerate. How many carbon 1,3-bisphosphoglycerate? 3 carbon. Now there is no change in the carbon number. What has happened? No. You remember the same step, recall it. Just now we read in the C3 cycle. What is the first stable product? G3P. It is converted into 3PGA. You remember what happened over there? Reduction. Same thing happening here. Glyceraldehyde 3-phosphate reduced to 1,3-bisphosphoglycerate. Correct now? It is what reaction? Reduction. So this is the most important step. Since it is a reduction reaction, 1 NAD reduced to NADH2. There is only one reduction reaction happens in the glycolysis, that too in the 6th step. So when 1 glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate, we produce 1 NADH2. But the same step we should also consider for dihydroxyacetone phosphate. So in the 6th step, how many NADH we produce? We produce 2. Correct? Now while calculating, always calculate for 2. For easy understanding, understand for 1. 1 glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate, we produce 1 NADH2. Similarly, dihydroxyacetone phosphate also converted into 1,3-bisphosphoglycerate. There also there is a production of 1 more NADH2. Hence, in the 6th step, we produce 2 NADH2 molecules. Then, in the 7th step, 1,3-bisphosphoglycerate converted into 3-phosphoglycerate. What's the difference between 1,3 and 3 here? 1,3-bisphosphoglycerate is how many carbon? 3 carbon. And 3-phosphoglycerate is how many carbon? 3 carbon. There is no change in the carbon number. What happened now? So the phosphate group present at the first carbon released out. Correct na? That's why 1,3-bisphosphoglycerate converted into the 3-phosphoglycerate. So it lost its phosphate or released its phosphate. So someone definitely accept that phosphate. Who will accept? ADP. Combines with phosphate, then there is a production of ATP. This is for one molecule I have considered. I should also consider for dihydroxyacetone phosphate. So in the 7th step, how many ATP I will get? Definitely I will get 2 ATP. Correct na? Now 3-phosphoglycerate is how many carbon? Converted into 2-phosphoglycerate. 2-phosphoglycerate is how many carbon? 3 carbon. Then what happened? So the phosphate group present at the 3rd carbon shifted it to 2nd carbon. Always remember, whenever there is a shifting is happening, the enzyme always involved is mutase. Okay, enzyme involved is mutase. So the phosphate group present at the 3rd carbon shifted to the 2nd carbon. There's no change in the carbon number. Now 2-phosphoglycerate converted into phosphoenol pyruvate, and 2-phosphoglycerate is 3 carbon. Phosphoenol pyruvate is 3 carbon only. There is no change in the number, carbon number. But what happened? It is a condensation reaction where there is a loss of water molecule. How many water total? 2 molecules of water. Correct na? 2-phosphoglycerate converted into phosphoenol pyruvate, 1 water molecule released. DHAP converted into phosphoenol pyruvate, again 1 more water molecule will be released. And what is the final step? Phosphoenol pyruvate converted into pyruvate. What happened over there? Phosphoenol pyruvate just lost its phosphate. Then ADP combines with PA, then there is another ATP. But since we have to consider for 2 molecules, so in the 10th step we get 2 ATPs, 7th step we get 2 ATPs, 6th step we get 2 NADH. Now once again quickly recall our C4 pathway. In the C4 pathway, what we discussed, you remember? Phosphoenol pyruvate, if it wants to be converted into pyruvate, there we used how many enzymes? How many ATP? 2. Remember, why the enzyme is dikinase. But here we are using only one ATP molecule. Okay, I'll quickly summarize, whomever did not understand, focus. Okay, focus everyone. So we have glyceraldehyde 3-phosphate in the 6th step converted into 1,3 bisphosphoglycerate. What reaction it is? Reduction. See, I'll simply tell you, always remember the statements. Wherever you come across the production of NADH and FADH2, remember one common statement. Enzyme always involved is dehydrogenase. Always. Okay. So what we have done in the 6th step? Glyceraldehyde 3-phosphate converted into 1,3 bisphosphoglycerate. So the enzyme is what? Dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. So we produce a total how many NADH? 2. Correct na? Here I am not writing for DHAP, but you have to consider that. Now 1,3 bisphosphoglycerate, what happened? It lost its phosphate group. Then that can be gained by the ADP and there is a production of ATP. Correct na? Then what do we got? 3-phosphoglycerate. 3-phosphoglycerate converted into, so phosphate group from the 3rd carbon shifting to the 2nd carbon. Now 2-phosphoglycerate converted into phosphoenol pyruvate. Phosphoenol pyruvate lost the phosphate group, converted into the pyruvate. So by the versatile enzyme pyruvate kinase. Okay. See, even 10th, 11th standard students also will read this much easily. Okay. So 90% we don't get the questions from here. Okay. We get the questions only from the regulations, which is very tough. We have to understand correctly. Okay. Now let's quickly understand the stoichiometry. So if you look into the stoichiometry in the step 1 and step 3, how much we invested? Step 1 we invested 1 ATP, step 3 we invested 1 more ATP. So we invested total 2 ATP. And which steps we gain ATP? Step 7 we got how many? 2. Step 10? So total we got 4 ATP. Correct na? So we spent 2 ATP, that's why I am subtracting. Then what is the net ATP? So the net ATP definitely will be only 2 ATP for one oxidation of glucose molecule only in the glycolysis. Correct na? But we also discussed in the 6th step of the glycolysis, how many NADH we got? 2 NADH. We all know very well 1 NADH is going to give rise to minimum of 3 ATP. Correct na? So 2 NADH give rise to? So 6 plus 2, 8. 8 ATP is called gross. Okay. Now don't get confused. Suppose if I show you 8 rupees check, okay, does it indicate I have direct cash in my hand? No. I have to go to bank, I have to encash it. Correct na? But if I show you 2 rupees coin, what does it indicate? I have a net amount. Similarly, how many ATP you can see with the naked eye? 2. What you can't see? 6 ATP. Why? Because this NADH has to enter into the electron transport chain. There it gives rise to this ATP. So total represents what? Gross. So gross 6 plus 2, 8. Total available. But you can see only 2. That's what it is called net. Correct na? There is a difference between the gross and net. Okay. Understand that correctly. Okay. Okay, just 10 minutes, we'll continue it. So consider we don't get the question from simple glycolysis, but in the exams like a SET and JAM, there is a possibility. Now, majorly we all has to focus on the very important part that is called as regulation part. Okay. So as I said, right, every question comes from the regulations itself. Okay, why it is not visible? Okay. So how much excited we are in the first day, you all people has to be excited till the end. That is called real preparation. Okay. Consistency is the real key. Intelligence is just 1 or 2% is required. Okay. Okay. How many of you want to crack the exam in this December for the CET and NET? Raise the hands. Others? Okay. Okay. Others? See, keeping a great aim is very important. Our effort also should be very higher. Okay. The more effort we put, more easily we will qualify the exam. Okay. Think about only effort, because efforts are only in our hands. Neither success nor failure, nothing is in our hand. This is not rocket science exam, at the same time not that much easy exam also. Okay. You require brilliant understanding and analysis. Okay. Just read with me 65 days without giving up. Okay. We can hope for the very better result. Okay. So please increase the time period at least 6 hours, 7 hours or 8 hours continuously. Then only we can cover the whole syllabus. Otherwise it's very difficult. Glycolysis regulation. Just a minute. All right, okay. Now let's continue. Whatever we discussed so far is the fate of the glucose. Finally, glucose has given the pyruvate, and that pyruvate, depending on the presence or availability of the oxygen, it is entering into the mitochondria. Okay. Before entering into the mitochondria, we have to understand how this glycolysis is regulated. In other words, instead of using the word regulation, we can use the word how it is getting operated. Okay, this is only the toughest part. If you understand this, believe you can answer one question definitely in the exam. Okay, focus on the concepts correctly. Okay, see, regulation of glycolysis is under the control of 3 enzymes: reaction 1, reaction 3, and reaction 10. You tell me what are those enzymes? Reaction 1 catalyzed by hexokinase. Reaction 3 by phosphofructokinase PFK. Reaction 10 is called as pyruvate kinase. Correct na? So there are 3 regulatory enzymes, and apart from the 3 regulatory enzymes, we also use an important line. Remember this, we use the word rate limiting enzyme. What is the difference between regulatory enzyme and rate limiting enzyme? In very simple words, regulatory enzyme functions can be replaced, but rate limiting enzymes functions cannot be replaced. Let's try to understand this correctly. What's the function of hexokinase? Conversion of glucose to glucose 6-phosphate. Correct na? You imagine a condition, hexokinase is absent. Hexokinase is absent. Then, even though in the absence of hexokinase, we can easily synthesize glucose 6-phosphate. How is that possible? We read right a mechanism called glycogenolysis. What is the glycogenolysis? Breakdown of glycogen. When I break down the glycogen, I will get glucose 6-phosphate. Again, I can produce glucose. So what is the point? Even in the absence of this enzyme, I can produce glucose 6-phosphate. Hence, it is a regulatory enzyme but not rate limiting enzyme. Now look into this most important one. Pyruvate kinase function is what? Conversion of phosphoenol pyruvate to pyruvate. Now imagine pyruvate kinase enzyme is absent. Even though it is absent, we can easily produce the pyruvate from amino acids. And very, very good. What is that amino acid? Alanine. This is the question in 2023 paper. Okay. Alanine is the name of the amino acid which undergo a mechanism called transamination and give rise to pyruvate. Hence, it is a regulatory enzyme but not rate limiting enzyme. What is the function of phosphofructokinase? Conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. So if phosphofructokinase enzyme is absent, no one can synthesize fructose 1,6-bisphosphate. That's the reason it is the only enzyme can act like rate limiting enzyme also and regulatory enzyme also. That is the only enzyme. We have to understand in a very deeper way. Okay. So In case of energy equation, we already seen that we gain total 4 ATP, we spent 2 ATP, correct now? So the net gain is definitely only 2 ATP, okay. Regulation we'll discuss in the next class, you will get confused now, okay. Now, answer this slowly. The best way to answer is make it simple. The more deep you think, nothing you will get. Okay, always make it simple. Look at the question. In glycolysis, what compounds provide phosphate groups for the production of ATP? Firstly, in which steps we gain ATP? 10th step, what happens? Phosphoenol pyruvate converted into pyruvate. So that is the compound. That is the compound which is acting as a donor of phosphate group, which is involved in the production of ATP. What is the 7th step? 1,3 bisphosphoglycerate converted into 3 phosphoglycerate. So who is the donor? 1,3 bisphosphoglycerate. Correct na? So 1,3 bisphosphoglycerate and phosphoenol pyruvate. Okay, it is simple. Now all of you understand. Again, this is also a question in the 2022, 2024 and often repeated. Now we produced pyruvate, correct na? Pyruvate is how many carbon? How many pyruvates we produced? 2 pyruvates. Now 2 pyruvates, in the presence of oxygen, it is converted into how acetyl-CoA is how many carbon? 1, 2. Correct now? If 1 pyruvate giving rise to 1 acetyl-CoA, how many 2 pyruvates give rise to 2 acetyl-CoA? How many carbons are left over? 2 carbon. 1 pyruvate is a 3 carbon which is giving rise to 1 2 carbon containing compound left with 1 carbon released in the form of carbon dioxide. So we have 2 pyruvates giving rise to 2 acetyl-CoA. There is a release of 2 carbon dioxide. Correct na? This is all happening where? In the presence of oxygen. Now the most important step, this is a reduction reaction. In the reduction reaction, what do we get? Either NADH or FADH2 we get. Since it is a reduction reaction, what is the enzyme I told you? Dehydrogenase. Very good. What we are reducing? That's why it is called pyruvate dehydrogenase. So pyruvate dehydrogenate into acetyl-CoA. There is a release of 2 carbon dioxide and there is a release of 2 NADH. So when the pyruvate converting into acetyl-CoA also we get 6 ATP, but not a direct form, we get 2 NADH. And this is only happening where? In the presence of oxygen. Correct na? Is it a carboxylation or decarboxylation? This reaction. Slow. How it is? What is called carboxylation? Addition of carbon. Decarboxylation is removal. See, that is the simple line they have given in the exam. Okay, we have to remember. See, because we are removing the carbon. Correct na? Because 3 carbon converted into 2 carbon. There is a loss or release of 1 carbon. So that's why pyruvate converted into acetyl-CoA by decarboxylation process. Don't confuse. This is in the presence of oxygen. Now imagine in the absence of oxygen. Whenever we are doing a vigorous exercise, running, cycling, all of a sudden muscle starts cramp. Correct na? Why? Because pyruvate is replaced by lactic acid. When we take a deep breath and we sit quietly for some time, again lactate replaced by the pyruvate. So pyruvate is how many carbon? 3 carbon. Very good. Lactate, it is also a 3 carbon. Hence, is there any release of carbon dioxide? Definitely there is no release of carbon dioxide. So pyruvate converted into lactate. It is also a reduction reaction. Okay. Now don't here, I'm so sorry, it is not a reduction, it is an oxidation reaction. Not reduction it is. Okay. Pyruvate converted into lactate by an enzyme lactate dehydrogenase. Dehydrogenases are the enzymes which perform redox reactions, both oxidation and reduction. No carbon dioxide release and there is no production of energy. Correct or not? When muscles are cramped, we don't produce any energy. Correct na? In fact, we lost energy. right? Now this is in case of muscles. What about in case of a yeast? In case of yeast, pyruvate converted into ethanol, correct now? In the absence of oxygen, pyruvate is how many carbon? 3 carbon. Ethanol, 2 carbon. Again, definitely there is a release of carbon dioxide. It is also a oxidation, okay, not a reduction. So there is only one reduction reaction. So we can observe this picture. Pyruvate in the presence of oxygen converted into acetyl-CoA, production of NADH2, release of carbon dioxide. Only one reduction. Now under anaerobic condition, in case of a skeletal muscle, it is converted into lactate and it is undergoing oxidation. NADH2 break down into NAD+. Is there any release of carbon dioxide? No. Then pyruvate in case of a yeast, in the absence of oxygen, converted into ethanol. Again, ethanol is how many carbon? 2 carbon. Again there is a loss of a release of 1 carbon. I may say 2 carbons. And here again it is what reaction? Oxidation. So there is only one reduction reaction, that is when pyruvate is converted into the acetyl-CoA. There is no need to get confused. Okay. Answer that. What about others? Still people has to come now? Still enjoying. Where is this Supratik? He didn't come now. I think he'll come. Online? What's the answer? Online? Reactions that converts glucose to pyruvate, call it as glycolysis. Simple. Okay. Metabolic reactions that break down large molecules to smaller molecules plus release of, what is this energy here? So what reaction it is? Catabolism. Correct na? Produced during anaerobic condition is lactate. So remember, coenzymes. NADH is a coenzyme. FADH2 is coenzyme. Correct now? What is the function? Transfer of electrons or the protons. That's all. Substances that remove or add H atoms in oxidation and reduction reaction, they are said to be the coenzymes. Correct no? Alright, everyone is clear. Okay. So we'll continue in the next class. Okay. From the next Monday, I told you, right, 1 to 2 will have lunch break. Okay. 2 to 3:45 or 4 I will take the class, followed by 1 hour of study hour. Okay. And every Saturday we will do the PYQ session. Okay. Of a whole week, and every Sunday will be the exam. Okay. This 60 days work very, very hard. Okay. From tomorrow all the classes will happen through the app only. I'll share the link in the new batch.